The authors have no financial interests in the subject matter of this film. Well, I was hit by a disease, keratoconus. Then my patient asked me, why? This video is about why. Though we have a variety of treatment options, do we actually understand keratoconus? Treating a disease without knowing why and how is a sin. Is this the only clue we have? The answer is no. Behavior of the eye is changing. We, as clinicians, have to find a logical theory of the disease right into the gene and molecular level. Optimal nurturing of the environment will maintain health, and break in this chain will result in pathology. We humans have a tendency to build everything based on a model. Hence, we propose a model for understanding the pathogenesis of the disease and pave the way for newer and effective therapeutic strategies. We do not discard anything we remove. Instead, we preserve them in our biorepository at Grow Laboratories and analyze them for studying various molecular and genetic markers. Let's rewind. Genetic and epigenetic contribution to pathogenesis is well known for a variety of diseases, and keratoconus is no exception. Mutations and genetic variations can influence collagen remodeling and possibly eye rubbing in keratoconus patients. It can cause classic keratoconus disease molecular phenotype, that is, low LOX and collagen synthesis and increased collagen degradation. And we have reported such a mutation in the VSX1 gene in keratoconus patients. Epigenetic trigger factors incite a cellular response with the production of inflammatory molecules such as cytokines and matrix metalloproteases. This results in loss of epithelial barrier function, lower LOX, and unfavorable collagen remodeling. This was reported by us as well. Cyclosporin can be effective in stalling the disease progression, and its preoperative use can reduce postoperative complications. We were inspired by Professor Ushumi's Nobel Prize-winning cellular phenomenon of autophagy, a process every cell has for maintaining its health by self-eating excess intracellular material. Our study did reveal that this process was dysregulated in keratoconus. Hence, modulating autophagy process has emerged as a promising strategy in the management of keratoconus. An important trigger for eye rubbing is the presence of itch factors. IgE is known to facilitate the release of itch factors that could cause eye rubbing. Its increase in serum and tear fluid in keratoconus patients implicates its causal role. Treat ocular allergy. Get a systemic workup done. If IgE is high, get an immunologist opinion and a patch test done. Get treatment for avoidance and desensitization of allergens. Hence, managing local and systemic IgE levels will be beneficial for treating keratoconus. Appreciating the concepts of nature and nurture in keratoconus will have a positive impact in the clinics. Genetic and epigenetic factors influence preoperative, intraoperative, and postoperative complications. Addressing them has strengthened our preventive and management strategies, improving treatment outcomes. Treatment in future may involve collecting tears, analyzing inflammation and other biomarkers, and treating for those specific markers, and also correcting defective disease-causing genes. This model, nature and nurture, is our work in progress, an attempt to understand the disease better, which will pave new roads in early prediction of disease and possibly halt surgical intervention for a better visual outcome and quality of life.